So it's a recording for CBT session 2017. The first question is regarding cytoplasmic mRNA. Cytoplasmic mRNA is the mRNA which is like coming to cytosol after post-transcriptional modification of hnRNA, right? So hnRNA is a precursor molecule that's modified by post-transcriptional modification and it's converted to mRNA. It's a nuclear process. The post-transcriptional modification of heteronuclear RNA, which is a very large molecule, is a post-transcriptional modification which is taking place in the nucleus of the cell. So in that process, when we see the size of hnRNA reduces in an attempt to form final mRNA, the size reduces. This is due to the fact that there are number of exon and introns which are alternatively arranged in hnRNA, and during splicing number of introns are actually spliced out. Introns are removed and exon ends are united together. So that will reduce the size of this RNA, and this final mRNA, if you see, it has got majority of exon in it. Intron numbers are limited, they are less in number. So size reduces. When size reduces, even the molecular weight reduces. Now when we see this particular question, regarding cytosolic mRNA, it's chiefly translated from nuclear DNA. In fact, it should be transcribed, right? It's chiefly transcribed from nuclear DNA. So true only. mRNA is like mainly coming from genomic DNA, that is nuclear DNA. Some amount of mRNA we certainly get in mitochondria, which is like transcribed from mitochondrial DNA, but chiefly transcribed from nuclear DNA. It's a true statement. So this is going to be the answer in this. Other options are all wrong statements, because the sugar cannot be deoxyribose in any RNA. RNA means ribonucleotide. You have ribose in RNA. Deoxyribonucleotide is DNA component. There you find the deoxyribose. So this is a wrong statement as far as RNA is concerned. Thymine is characteristic base which is found in DNA only, not in RNA. So this is also a wrong statement. Molecular weight of mRNA greater than hnRNA is also the wrong statement, because you have seen the size of mRNA is reducing compared to hnRNA. So the molecular weight also will reduce compared to hnRNA. Now coming to second question. Now all of the following type of porphyria are autosomal dominant except. It's a very important question. All hereditary porphyrias are autosomal dominant, except CEP, which is autosomal recessive. CEP is congenital erythropoietic porphyria. Otherwise, when you talk about AIP, PCT, HCP, VP and PP, they all are autosomal dominant in nature. CEP is autosomal recessive in nature. And there is one acquired porphyria, which is called plumboporphyria. Plumboporphyria. This is an acquired porphyria, which is lead-induced porphyria. So lead-induced porphyria is acquired porphyria, and then this is a purely neurological porphyria. Purely neurological symptoms the baby will be having, like abdominal pain and other features. So it's a lead-induced porphyria, and this is due to inhibition of ALA dehydratase enzyme of heme synthesis. ALA dehydratase enzyme. The lead is going to block this enzyme activity that will lead to accumulation of lot of ALA which will enter the brain cell. That will lead to plumboporphyria. Now the first step of liver metabolism of fructose is, this is wrong. Phosphorylation to fructose 1,6-bisphosphate, this is also wrong. Fructose 6-phosphate is wrong. Fructose 1-phosphate is true. When we are talking about fructose metabolism in liver, we see That first enzyme which is acting on fructose is fructokinase, and it's a specific enzyme for fructose which converts the fructose to fructose 1-phosphate, not the 6-phosphate. And this fructose 1-phosphate, when it is produced, it is cleaved to DHAP and glyceraldehyde. DHAP and glyceraldehyde. So this fructose 1-phosphate is going to cleave into two, DHAP and glyceraldehyde. Because the substrate is having only single phosphate, that phosphate is going to go to DHAP and glyceraldehyde will be left, poor glyceraldehyde will be left without any phosphate. And this breakdown is by aldolase B. This will enter the glycolysis directly, and this glyceraldehyde also may enter the glycolysis, but only once it is converted to, after it is converted to glyceraldehyde 3-phosphate, it can be utilized in glycolysis. So this fructose 1-phosphate is breaking into glycolysis and breaking down finally in the glycolysis by these particular process. This you remember. Now the first step it is asking. Obviously it is fructokinase step, and that fructokinase is going to phosphorylate the fructose to fructose 1-phosphate by the expenditure of ATP. Of course you have expenditure of ATP at this point, right? So this is the thing about the fructose metabolism. Now cabbage odor urine is the characteristic feature of tyrosinemia type 1. You have type 1, type 2, type 3 tyrosinemias. Type 1 is having the odor, and that is cabbage odor. Type 2 is odorless, type 3 is also odorless. So only type 1 is having the problem of this cabbage odor urine, and in this tyrosinemia type 1 you have deficiency of hydrolase enzyme, which is going to convert the fumaryl acetoacetic acid to fumaric acid and acetoacetic acid. That is the terminal enzyme for catabolism of fructose, that is deficient in type 1. And if they ask you, this also very frequent question, type 2 tyrosinemia, you have deficiency of first enzyme of tyrosine degradation, and that is tyrosine transaminase enzyme. This is tyrosinemia type 2. Tyrosine transaminase deficiency. Coming to next question. When you talk, like if you see this question, I feel that this question is having one more important odor in the urine. Leucine catabolism defect. You get isovaleric acidemia, and this isovaleric acidemia will be called isovaleric aciduria when you find the isovaleric acid in the urine, and that's giving you cheesy odor urine. So don't get confused between these two terminology, the cabbage odor and cheesy odor. It's a different. Cabbage odor is tyrosinemia type 1 and cheesy odor is isovaleric aciduria, which is due to leucine catabolism defect. Now predominant form of LDH in the cardiac muscle, it is type 1. See, LDH is having 5 isoenzyme. You have LDH1, LDH2, 3, 4, 5. LDH1 is having all 4 subunit as H type. LDH5 is having all 4 subunit as M type. 2 is having 3 H and 1 M. 3 is having 2 H and 2 M. 4 is having 1 H and 3 M. And 5 is having all 4 as M subunit. So if you talk about LDH1, all 4 subunits are heart type. M means muscle type, H means heart type. So all 4 subunits are heart type subunits, and this is the form which is predominantly found in the heart muscle. When you see the plasma, the LDH2 is more than LDH1. This is the rule. In the normal plasma, 2 is more than 1. But in case of MI, myocardial infarction, what happens? The LDH1 fraction increases in the plasma, that increases the LDH1 more than LDH2. This is called flipping pattern, flipped pattern of LDH, flipped pattern of LDH, flipped pattern of LDH. Where LDH1 becomes more than LDH2, it's called flipped pattern of LDH, and this is seen in MI. Flipped pattern of LDH which is seen in MI. So this you should know. So predominant one which is found in the heart muscle is LDH. So this is also actually kya hai? This LDH actually increases little slowly in the plasma after MI. Takes around 2 to 3 days to raise in the plasma, but it lasts long. Say for example, it lasts for 10 days. So it's like late marker of myocardial infarction. The time when other markers have been there but have disappeared from the plasma, this LDH may act as a reliable marker to find out the MI in the remote past. So it is going to last for 10 days in the plasma. And when you talk about CPK and all, they will last only for 72 hours or so. So if you want to find out whether the person was having a MI a week before or not, you can use this particular marker as a MI marker. Now coming to the next question, amino acyl tRNA is required for all except. This is the transfer RNA, basically, which is going to carry the amino acid to the point of translation. So we have 61 codons which are meant for 20 different amino acids. And how 61 codons are coding for only 20 amino acid? The answer is degeneracy. So 61 codons coding for 20 amino acid. This is because of degeneracy phenomena. And you should know one thing, that to transfer these 20 different amino acid, you have more than 20 types of transfer RNA. You have 31 types of transfer RNA in the cytosol. For the nuclear genome, you have got 31 types of transfer RNA. So one amino acid is carried by more than one type of tRNA, you should know. The standard amino acid which we are talking, like valine, leucine, isoleucine, alanine, histidine, proline, all these amino acids, they have their dedicated RNAs, transfer RNAs to transport them. So is required for all of the following except. So for this also, you have transfer RNA which can transport these respective amino acid to the site of translation. But when you're talking about hydroxyproline, you understand, hydroxyproline is actually the amino acid which is generated from proline after post-translational modification of the proline residue. So hydroxyproline, if it is there in the polypeptide, it is not inserted there during the translation process. Rather, it was actually the proline which is modified post-translationally to hydroxyproline by prolyl hydroxylase enzyme. So this occurrence of hydroxyproline or hydroxylysine is actually a post-translational modification. So they are never carried, at the site of translation, and they are not directly getting incorporated in the polypeptide because none of the codon is coding for hydroxyproline as such. So this hydroxyproline is actually never carried by tRNA. Same applies for hydroxylysine also. So this particular statement is the answer. Hydroxyproline is not having any transfer RNA. It is more interesting if they ask you what is the transfer RNA for carrying the selenocysteine. This is 21st amino acid and this is a modified form of the serine. You should know that it's not derived from cysteine. Rather, the serine is the precursor molecule. Serine is a hydroxyl group containing amino acid, and this serine oxygen is replaced by enzyme called selenocysteine synthetase. And that place of oxygen you get to have selenium. So actually this serine is modified to selenocysteine. and this modification takes place at the time when the serine is already charged on its respective tRNA, and it's going to, this is 3 prime, this is 5 prime, and you understand this is tRNA. Now this is the serine, happily it was sitting here at 3 prime end, but the enzyme came and modified the serine to produce you selenocysteine. So if they ask you which transfer RNA, what is the transfer RNA which is transporting selenocysteine, you have to say it is the transfer RNA meant to carry serine. Transfer RNA of serine actually is responsible for transportation of selenocysteine at the point of translation. So selenocysteine is carried by serine transfer RNA to the point of protein synthesis. And one more thing, the occurrence of selenocysteine in the polypeptide, it's not opposed to translational modification, rather it is a co-translational process. Means when the other amino acids are being incorporated in the polypeptide, that time itself some of the yugas, they start coding for selenocysteine and that's getting incorporated there. So occurrence of selenocysteine in the polypeptide is not a post-translational modification like we have seen for hydroxyproline and hydroxylysine, which are there due to a post-translational modification. The occurrence of selenocysteine is rather big, it's a co-translation process where already the serine is converted to selenocysteine at the time when it is being transported by tRNA, and then when the selenocysteine is formed, that's getting incorporated in the polypeptide at the time of translation itself. Now modification of nucleotide of the pre-transfer RNA, rRNA and pre-mRNA occurs. Obviously occurs post-transcriptionally because already they have been produced, they are transcribed. And when they are modified, it's called post-transcriptional modification. So answer is going to be post-transcriptionally. When the polypeptide, the protein is getting modified after synthesis, it's called post-translational modification, and this is called post-transcriptional modification, right? Now next to this, question number 18, ubiquitin, breaking down the protein in proteasomes. This is a protein. This protein is a special protein because this protein is having short half-life, or this is a kind of protein which is having the regulatory role, like for example certain enzymes and factors which are having effect on regulation of certain metabolic pathways. So such proteins, when they have completed their function, they need to be degraded. So for that, number of ubiquitin residues, this is all ubiquitin. So number of ubiquitin residues will be added at one of its tail, one of its end, and this is called ubiquitation of the protein. Ubiquitin itself is a 76 amino acid long protein. Ubiquitin itself is a 76 amino acid long protein. And this ubiquitation of protein will carry this protein to a organelle which is hollow inside and having 7 layers of proteins. Each layer is made up of 4 subunits. So you have total 28 subunit in this kind of a structure which is cytosolic, and this structure is called proteasome. So this proteasome is the hollow organelle where this kind of ubiquitinated protein is going to go and that's undergoing degradation. This is called proteasome structure. So this you should know. Now coming to question number 9. The reading of the codon on the mRNA, it's 5:3. Contrary to the fact that reading of the DNA and reading of the RNA, reading of the DNA for DNA synthesis and reading of the DNA for RNA synthesis is occurring in 3 to 5 directions. Whenever you're talking about reading of the nucleotide at the level of DNA, the reading at the level of DNA will be 3 to 5. But when you are talking about the RNA reading, the codon reading on mRNA, the reading will be there in 5 to 3 direction. That you should know. This is mRNA, 5' end, 3' end, and you have codon like this. And this is how like the ribosome is occupying the position on mRNA. This is the transfer RNA having methionine on it. It's an initiator amino acid. So this methionine, this is a methionine amino acid. So this methionine, if you're thinking that it's identifying, like this anticodon is identifying the codon and methionine codon is AUG, you understand we are reading AUG in 5 to 3 direction over here. So AUG. Similarly, if you say UGA, we are reading this UGA nucleotides in 5 to 3 direction. This will act as a stop codon. We are not reading it as AGU, right? So we are reading it as UGA and we are telling it's a stop codon. So reading on the mRNA during translation process is always taking place in 5 to 3 direction. This you remember. So this question is quite simple question, but it's having a lot of depth in this, right? So you know that whenever we are talking about the DNA replication, when the DNA is read and daughter DNA is produced, the reading of nucleotide on the DNA is 3 to 5 direction. And similar fashion, when the nucleotides are read on the DNA in some area or a gene, the reading again is going to be in 3 to 5 direction, and daughter RNA or daughter DNA production is going to be there in 5 to 3 direction. Production is going to be there in 5 to 3 direction. And when we are talking about mRNA, as I have just now told you, the reading of codon will be in 5 to 3 direction, and the polypeptide synthesis will be there in N to C terminal end. Polypeptide, the protein, we are calling it as N terminal or C terminal, and the direction of the synthesis of polypeptide will be from N terminal towards C terminal end. Next to this, we will see 10th question. The first reaction involved in the degradation of most amino acid involves the participation of transaminase enzyme, where the toxic amino group of the amino acid is transferred to the keto acid. Majority of the enzyme, I'm not telling all, but majority of the enzyme, they follow this route as a first reaction of their catabolism, where the amino group of amino acid is transferred to a keto acid. In most of the circumstances, that keto acid is going to be alpha ketoglutarate, which is intermediate of TCA cycle. This converts the amino acid to respective keto acid, and this keto acid is converted to, this alpha ketoglutarate is converted to glutamate. This reaction is catalyzed by transaminase enzyme, and this transaminase enzyme demands presence of B6 for activity. B6 PLP. PLP is pyridoxal phosphate. It's active form of vitamin B6. So transaminase needs B6. Transaminase needs B6 enzyme. So first reaction involves, for most of the amino acid, this particular cofactor for activity of transaminase. Coming to 11th question, the statement which is not correct. Side chain of cysteine and methionine absorb light at 200 nanometer. This is the answer, because this is not a correct statement. At 280 nanometer, if you're talking about the absorption of ultraviolet light, that's done only by aromatic amino acid, that is 3 number. One is tryptophan, tyrosine, and phenylalanine. Tryptophan, tyrosine and phenylalanine. Maximum absorption will be seen for tryptophan. Least absorption of this light will be seen for phenylalanine. Cysteine and methionine, they do not show the absorption. So this is the answer. All other statements are correct statement. The glycine is often present at a region where polypeptide is forming a sharp bend, reversing the direction of the polypeptide. This statement is talking to you about beta bend. There are beta sheets. This is like polypeptide and terminal C-terminal. This is folding on itself. This is folding on itself in this particular fashion. It's called pleating. It's called beta pleated sheet. It's called beta pleated sheet. This area is called bend, beta bend or beta turn. This area is called beta bend or beta turn. This is, it's a very narrow area, so it will accommodate the amino acid which are very small in size like proline and glycine. So this is the area which is having especially rich amount of glycine and proline. So this is a true statement. Now C, N, H and O, atom of the peptide bond is coplanar. This is also true. They are all lying on a single plane. When we talk about peptide bond, we have got contribution of all these 4 atoms and they all are coplanar in nature. And so this is true. And this is also the true statement. The pentapeptide. Pentapeptide means the number of amino acid is 5. Pentapeptide means amino acid number is 5. So when you have 5 amino acid, 1, 2, 3, 4, 5 amino acid, they are linked by peptide bond. And if you count the number of peptide bond, you have 1, 2, 3, 4. 4 peptide bond which are linking 5 amino acids together. So it's having 4 peptide bonds, also a true statement. So answer for this particular question which is not correct is the A, because cysteine, methionine, they do not have. It's a very important question. It's a very nice question and each statement is an MCQ in this. Now which of the following is not correct? Metabolic disorders are infrequently associated with catabolism of purine. Not correct. This is not correct. This is the answer in this question. Because purine catabolism is associated with number of disorders. You have severe combined immunodeficiency. You have immune dysfunction where only T cells are affected. B cell normal. Severe combined immunodeficiency means the T cell as well as B cell dysfunction is there. When you talk about immune dysfunction, it's another enzyme disorder where you have only the cell mediated immunity not being there. Then you have xanthin urea associated with this, and they say that Lesch-Nyhan syndrome also, you have deficiency of HGPRT which will lead to hyperuricemia. Though you should know that Lesch-Nyhan syndrome HGPRT deficiency is not the metabolic defect of purine catabolism as such, because HGPRT enzyme is a salvage pathway enzyme. So actually the salvage pathway is getting affected in Lesch-Nyhan syndrome, and so there is excessive catabolism of hypoxanthin to form your uric acid, but you get to have hyperuricemia because of purine catabolism, right? So there are number of disorders actually being associated with this purine catabolism, because intermediates are not water soluble and they start disturbing one or other pathways. On contrary, when you talk about the catabolism of pyrimidine nucleotide, they are not associated with metabolic disorder because their intermediate are water soluble. So pyrimidine catabolism is not associated with metabolic disorders, but purine catabolism is associated with number of metabolic disorders which I have enlisted. So this is the answer. So all other statements should be correct statement. They all are correct statement. Number immune dysfunction associated both with defective adenosine deaminase and purine nucleoside phosphorylase. A true statement. The one like adenosine deaminase deficiency is seen in severe combined immunodeficiency, and ADA is actually the enzyme for purine catabolism. And immune dysfunction, which I was talking to you, is because of this second enzyme. What is that? A purine nucleoside phosphorylase enzyme deficiency. That will lead to only cell-mediated immunity dysfunction. Xanthine lithiasis can be due to severe defect in xanthine oxidase. It's also true. Xanthine oxidase will lead to xanthinuria, and when xanthinuria is there, you get to have the stone, xanthine stone in the urine. Now, hyperuricemia can result from conditions such as cancer characterized by enhanced tissue over. This is also a true statement, because when enhanced turnover of the DNA and RNA is there, you get to have a lot of purine nucleotide, which after catabolism gives you uric acid. So not only increased turnover of the cell in cancer cell, but also during therapy, when the radiation is given or the chemotherapy is given, the cells are dying and their DNA, RNA, they are undergoing catabolism to give you hyperuricemia. So cancerous conditions or any conditions where you get high turnover of the tissue, or you get to have some radiotherapy or chemotherapy, there you have high uric acid in the plasma of the patient. The initiator transfer RNA is placed within within the active ATS initiation complex, which of the following three canonical site during protein synthesis? This I have already covered in a previous question, but again I can, for your convenience, I'll be drawing it again. This is mRNA having 5' end and 3' end, and when we talk about initiation complex, ATS initiation complex, this is ATS initiation complex, which is the complex made up of ribosome, mRNA, and also one tRNA. The transfer RNA, which is having the methionine in it. So and this is the starting transfer RNA. The acceptor site codon is actually waiting for one new tRNA to come and occupy this place. So the question is, at which of the three canonical site protein synthesis the initiator tRNA is placed? Of course, the answer is P site. T site. Most appropriate statement about isoenzyme. Different Km values are true statement. Most of the enzyme, if they are isoenzymes, like what is isoenzyme? Isoenzymes are distinct physical form of the same enzyme having different physical characteristics. Suppose, like I was discussing you in the previous question, the LDH is having 5 isoenzymes. The distribution is different. Their structure is different. You have seen the subunit association in LDH. So distribution is different. So you write down, this is the difference, physical characteristics which are going to be different in isoenzyme. Number one, distribution in as far as organ is concerned, they are distributed differently in organs. And even if the isoenzymes are found in same cell, same organ, the compartment may differ. Some may be cytosolic, some may be mitochondrial. For example, if you talk about malate dehydrogenase enzyme, you have cytosolic and mitochondrial isoenzyme of MDH. So in terms of distribution, in terms of structure, they are going to differ. In terms of substrate affinity, they are going to differ. Substrate affinity. In terms of Km values, they are going to differ. Km values. When substrate affinity is different, the Km value also is going to be different. The isoenzymes may differ on their electrophoretic mobility also. Some will move fast, some will move slow. So it is quite possible to separate them by using electrophoresis. So most appropriate is E. Different substrate is a wrong statement, because that's a characteristic feature of isoenzymes, that all the isoenzyme of one enzyme will have same biochemical reaction, where they act on same substrate and catalyze the same biochemical reaction, producing you same product in same fashion. It's only their affinity to that particular substrate differs. Some will have high affinity, some will have low affinity. Consist of multimeric complex. Not necessarily. It is not necessary that isoenzymes are multimeric. The monomer also can be having, monomeric enzyme also may be having isoenzyme forms. And this also is wrong. As I have told you, electrophoretic mobility is going to differ. Now coming to question 15. I think this is the last question of the CBT. The enzyme avidin inactivates biotin in the egg. If you talk about egg, the egg is having outer albumin portion and inner yolk portion. You have to remember the biotin is found in yolk, and A for avidin, albumin is having avidin. This is a thermolabile component, so thermolabile one, and it is seen that avidin is having a strong tendency to bind biotin. It binds biotin, and one avidin is capable of binding 4 biotin. One avidin is capable of binding 4 biotin. So they have strong affinity to each other. So it's a thermolabile, but when you boil the egg, the avidin is lost and biotin is available for GIT absorption. So the enzyme avidin inactivates biotin in the egg. So if you consume raw egg, the avidin will be there in the outer white portion of the egg, and that will bind with biotin, will not allow the biotin absorption. Right? And this fact is actually being exploited in designing ELISA technology, where streptavidin and biotin is actually being used to code the antigen and antibodies respectively. So with this, I have completed the discussion of CBT 1. Thank you.